move on to talk about absorption. So we're going to describe the fundamental mechanisms involved in the absorption of carbohydrate, protein, lipid digestion products, and we're going to explain how the mechanism of a lipid absorption is related to the non-polar nature of fats. So we're going to look at the mechanism by which the micronutrients, so remember carbs, proteins, lipids are macronutrients. And so when we ingest them in the way that we take them in our food, in our diet, they must be broken down to their smaller components. So carbs are going to have to be broken down to glucose, which are the monomers. Proteins are going to be broken down to amino acids. Lipids are going to be broken down to monoglycerides and fatty acids. And so we're going to talk about how we get these large macromolecules broken down to smaller macromolecules, smaller micromolecules, excuse me. And then we'll talk about how those smaller micromolecules or micronutrients can be absorbed across the epithelial cells of the GI. So we'll talk about carbs, proteins, lipids, and then we'll look at the way vitamins are absorbed, minerals, and then water. So this is going to call on us to remember or to recall the mechanisms of transport that we talked about last semester. So active transport, which is requiring the sodium-potassium pump, which we can see up here. And just to remind ourselves, this pump is pumping 3 sodium out and 2 potassium in. And so what this sodium-potassium pump is doing, and we saw this in the kidneys as well, is it's creating a deficit of sodium inside the cell. It's pumping sodium out, which is creating a gradient for sodium to move passively into the cell. So when we have this pump on the basolateral surface of our epithelial cells, we can then passively move sodium in on the apical surface of these epithelial cells. And as we move sodium passively in, this is an opportunity to use that energy to bring other products, other micronutrients, other molecules into the cell actively. Okay, so this is going to be secondary active transport, where we use the energy that's being liberated by one molecule, one ion, sodium, moving into the cell, and we can move other things into the cell, such as amino acids and glucose, galactose, etc. So this is one form of transport that is really evident here in the absorption of nutrients in the small intestine. Another form of transport we're going to see is facilitated diffusion. So facilitated diffusion is where we have a transporter. So it's also moving molecules with a transporter, but here we have a gradient. So here it's going to be passive movement. So this is a form of passive transport where we're moving a molecule according to a gradient, but we're still going to require a carrier or transporter. So these are just some of the mechanisms of transport that we should be remembering from last semester that are going to be required in terms of understanding how absorption takes place. So let's start out by looking at carbohydrates. So carbohydrate digestion, we said, starts in the mouth. Only monosaccharides can be absorbed. So we're going to have to get these carbs broken down to their smallest components. And that starts off with taking disaccharides and polysaccharides that are coming in from our diet, depending on what type of starch you've ingested. And then we break those down to monosaccharides, starting with salivary amylase in the mouth, and then pancreatic amylase in the duodenum, in the intestines. Now, the end products that we usually get are disaccharides, such as maltose, and limit dextrins. Limit dextrins are actually these short branched polysaccharides. And so when we get these products from salivary or pancreatic amylase, we have to further break them down into monosaccharides such as glucose or galactose, okay? So once we get them down to those smallest monomers, glucose or galactose, let's talk about how we actually move them across the small intestinal wall. so this is the process of absorption of carbohydrates. And so we're going to take one of our epithelial cells. So this is one of those cells that are lining the villi that we just looked at. And it's going to have these microvilli on top of it to increase the surface area. And we're going to have on the basolateral surface our sodium-potassium pump, which is pumping sodium out, potassium in. Okay, and because we have the pumping of this sodium out, we're going to create a deficit of sodium inside the cell. So therefore, on the apical surface, we can now move sodium into the cell passively. Okay, and as we move sodium into the cell passively, we can then bring another molecule. In this case, we're looking at things like glucose and galactose. Using the energy that's liberated there. So the transport that's happening here is active. I'm going to put this in red so that you can see that's active. Hopefully you can see that. The transport of sodium here is therefore going to be passive. And then we're going to be able to use that energy to bring glucose and galactose into the cell. Okay, so let's describe these mechanisms here briefly, just so you have an idea. So this is going to be the sodium-potassium pump. And we said that this was primary active transport. And this is the sodium-linked glucose transporter, which is secondary active transport. Okay. And remember here, this is going to require energy. The energy for this process is liberated by hydrolyzing ATP into ADP and a phosphate. So that's the energy for this process. Now, so we've gotten glucose and galactose into the cell, okay? Once we have glucose into the cell now, we're gonna start building up a gradient here again. So we're gonna have increased levels of glucose inside the cell. And so now across the basolateral surface via facilitated diffusion, we can then absorb that glucose and galactose into the underlying blood vessels. So let's draw those blood vessels here. So underneath this cell, we have our capillaries. And this is where we want that glucose and galactose to diffuse to. So it's gotten into the cell across the apical surface. Across the basolateral surface, we have a facilitated diffusion happening here. So we're going to be able to bring glucose or galactose across the basolateral surface using this facilitated diffusion as the mechanism. And I'm going to put this in green because that's going to be passive. Okay, I hope you can see those colors. Okay, so let's describe this second mechanism. This is going to be facilitated diffusion. Using a glucose transporter. Okay. And so the gradient here was that there's high glucose here, low glucose down here. And that is how we get that passive movement of glucose from the cell into the underlying capillary. Okay, so this is how we get glucose to move across the epithelial membrane, across the epithelial lining, and into the underlying capillaries of the intestines, of the small intestines. Okay, and so it's really just calling you to remember some of those transport mechanisms that we talked about. So primary active transport, secondary active transport, and then facilitated diffusion. All right. Okay, let me just pause here a little bit, see if there are any questions. Okay, so let's get back to this. So that was carbohydrate digestion and absorption. Let's move on to look at proteins. So proteins are going to be a little bit more extensive. We have lots of enzymes to break down proteins. They're broken down by a general class of enzymes called proteases. There are several types of proteases that cleave proteins in different ways. So we have endopeptidases that cleave polypeptides within the bonds, so in the interior of those peptide bonds. And then we have exopeptidases that cleave towards the ends of those peptide chains. Remember, the monomers that are making up proteins, that are making up these peptides, are amino acids. And when I say peptide, peptides are just smaller chunks of amino acids, basically smaller proteins. We also have zymogens. So zymogens are the inactive storage form of these proteases. So remember, our body's made mainly of proteins, right? The proteins that are active in our system, as well as our tissue that has proteins in it. And so we don't want these proteases to be active all the time. Otherwise, our body would literally digest itself. So they're in their inactive form when they're secreted, and these are what we call zymogens. Now, zymogens are pretty much storage granules, so granules that are stored inside these cells, and they're secreted via exocytosis, and they're activated via proteolysis. So there's one enzyme that is going to cleave one of the amino acids off of this zymogen, and that is what will activate it or turn it on, and then it can go ahead and break down the proteins that are in the small intestine. So these are all forms of proteases, and these are the different types based upon where they cleave, and we're going to look at the different names based upon what they cleave here on this slide. So protein digestion starts in the stomach. We talked about some of the cells of the gastric pit. So this is a gastric pit of the stomach mucosa. We have chief cells here. Chief cells secrete pepsinogen. Parietal cells secrete hydrochloric acid. And the hydrochloric acid that's secreted from parietal cells is required to activate pepsinogen to its active form, pepsin. Okay, so that's a part of the reason that we have this acid. It's one of the reasons that we have this acid in the stomach as well, to activate pepsinogen into its active form, pepsin. Now, when we have this pepsin activated, it can then cleave these larger proteins into smaller peptide fragments or smaller chains of amino acids. When we get to the small intestines, here we have these smaller chains of amino acids, or these smaller peptides, and here we have these pancreatic proteases that are released from the pancreas via the pancreatic duct, and here we can further break down these peptides into their smaller amino acid components. So there are two types of proteases here in the small intestine. They're the pancreatic proteases, which are trypsin, chymotrypsin, and carboxypeptidase. And then there are the brush border proteases, which are the aminopeptidase and the enterokinase. So basically, the pancreatic proteases are coming from the pancreas in the fluid, the pancreatic juices that are being secreted. When they get to the small intestine, the duodenum, they're going to be activated. First of all, trypsinogen is going to activate, is going to be activated by enterokinase. So enterokinase is one of the enzymes that is always found in the duodenum. So the brush border proteases are always in the duodenum. And they will then activate the pancreatic proteases that are coming from the pancreas. So enterokinase is going to activate trypsinogen into trypsin, and then trypsin will go on to activate the other pancreatic proteases into the activated form. So trypsin will activate chymotrypsinogen to chymotrypsin and procarboxypeptidase into carboxypeptidase. Trypsin will also inhibit more trypsinogen into trypsin as well. And so this is just a mechanism to make sure that these enzymes are only activated when they're in the small intestine. So the enzymes that are required to activate the pancreatic enzymes into their active form are only located in the intestine. This is preventing the pancreas from eating itself, right? The pancreas from activating these enzymes and digesting itself. So the absorption of amino acids, again, calls on these transport mechanisms, calls on us to remind ourselves of these transport mechanisms. So amino acids must cross the apical membrane by that same sodium-linked secondary active transport, or it can also use facilitated diffusion depending on the relevant concentrations of amino acids. And then across the basolateral surface, it's going to use facilitated diffusion, very similar to what we saw with glucose. Now, in order for amino acids to be absorbed, in order for amino acids to be absorbed, they must be in the form of amino acids, okay? So we cannot absorb dipeptides or tripeptides. They can enter the cell, but once they get inside the cell, they must be broken down to amino acids in order to cross the basolateral surface or the basolateral membrane, okay? So across the apical surface, we can absorb amino acids, we can absorb peptides and the longer chains of amino acids. But in order for them to cross that basolateral membrane into the capillary, they must be in their smallest component, which is amino acids.